Hello students, hope you are studying well this chapter, digestion and absorption. In the first video I had told that it's a big chapter, so I am doing small topics in every video. So let's start, and the today's topic, that is digestion of lipids. Human physiology is always very exciting and very interesting because we can always associate with it, right? So what will we do in digestion of lipids? We'll do the chemical digestion, right? We'll do the chemical digestion. We'll study about absorption, and we'll also study very little. We'll study very little about chylomicrons. When we'll study, then you'll know what are these chylomicrons, right? So this is all what we are going to do today, and it is a very interesting pathway that we are going to cover today. So now let's start. So basically, when we say digestion of lipids, what do we mean? I mean, what are we going to study? What are lipids? What are we going to do? So when we say that we are going to do digestion of lipids, we mean that whenever we eat, there is a big component of fat which we consume. Okay, so the big component of fat which we consume typically consists of kids, whenever I'm writing, please keep a notebook so that you are taking notes also. So what is it? What is this component that we are going to talk about? This is triglycerides, and this is the most significant which we are going to study from our point of view. It is most significant from our point of view. Then there is cholesterol ester, and then there are certain phospholipids. So this is the component. These are the components which we are going to study how they are going to be broken down, right? So when we say this triglyceride, cholesterol ester, and phospholipids, what are these? These are polymers, and we cannot absorb these. Our body cannot absorb these. So why? Because they are large molecules, and we know by the definition of digestion that large molecules have to be broken down into small molecules so as the body can absorb. So these polymers will be broken down into what? They'll be broken down into small molecules, right? They will be broken down into small molecules. And these polymers, when broken into small molecules, they are known as what? They are known as monomers. They are known as what? Monomers. So basically, what are we going to do today? We are going to break the polymers into monomers. And what is happening? Breakdown of polymers into monomers. And we can also say that it is a catabolic pathway. And or we can also say that hydrolysis is taking place. Whenever we say hydrolysis, what do we mean? We add water to the chemical reaction, right? So we are breaking down polymers into monomers. What polymers? Triglyceride, cholesterol ester, and phospholipids. Now let us see what will happen, right? So the first one that we had written was what? Triglyceride. This is a polymer. We cannot absorb it. So we'll break down. After breaking down, what will happen? It will become into fatty acids that a body can absorb, and some portion of monoacylglycerol, which is also known as MAG. In some books, this is not given, but When I'm telling you, I want to tell you the full things. The cholesterol ester, the ester bonds will be broken down and it will become cholesterol and phospholipids, right? Phospholipids. These were the three polymers that I told you. So, and these are the monomers in which they'll be broken down. So basically what happened, these are the polymers and these are the monomers. So these are the large molecules and which the body cannot absorb, and these are the small molecules which the body can absorb. Please remember this. Okay, now let us go further and see how it is happening. Before we go further, whenever we are saying breakdown, what is breaking down? Whenever we are saying breakdown, so what is breaking down, right? We are saying polymers into monomers. We are saying polymers to monomers breaking down. But what is breaking down, right? So basically, we'll be talking, when we are saying breaking down, these ester bonds are breaking down, right? These ester bonds are breaking down. So see, we have to study according to our syllabus, according to the questions that will be asked. But there are few things, very little things in which I'll go a little over and above the syllabus, very little, so that you can actually get a feel, you can actually get an understanding of what is actually happening. Okay, because see, if you don't understand, then there is no fun what is actually happening. So when I'm saying polymers to monomers breaking down, so we understood what is polymer, we understood what are monomers. Now the question is, something is getting broken. So what is getting broken? These ester bonds. And what are these ester bonds? Very little I'll tell you. This is not a biochemistry class. These are the carbon doubly bonded to oxygen, to a hydrogen, to a hydrocarbon, and to another hydrocarbon. So this is a ester bond which needs to be broken, right? Very little I have told you what are broken. So these all the enzymatic activity that takes place, that all the enzymatic activity that we will study will be breaking down what? It will be breaking what? Now you know that will be breaking ester bonds. Okay. Now let us see. We ate something that was fatty, that had lipids. So the first place where it goes is where it is at the oral cavity, right? Mouth, very in simple language, we eat mouth. So what is happening in the mouth? Remember I told you about salivary glands, right? Salivary glands. So you might be remembering there is one sublingual which is below tongue. You can see my video for salivary glands, right? If you want a detailed description, you can see salivary glands. So we had studied in this sublingual gland and parotid gland. Remember, in front of the masseter muscles, cheeks here, you remember the everything. Whenever I'm saying these words, that video, that everything should come into your mind. Then only it means you have studied nicely. So this sublingual and parotid gland, this will secrete what? It will secrete something into oral cavity. And what will be that thing? That thing will be lingual lipase. That will be what? That will be lingual lipase. Okay, and from the oral cavity, the food will go to where? To stomach. And in the stomach, what will be secreted? Gastric lipase, which will help in the breakdown of these bonds, ester bonds, which we have studied. Okay. Now let us see what is it, what is actually happening, how the food is getting, that fat is getting digested and absorbed, right? So we studied lingual lipase and lingual lipase. And what was the other thing that I said? That was the gastric lipase, right? So whenever I say this, these two will do what? Because it's hydrolysis, I told you. Suppose this is a molecule that needs to be digested. Okay, this is a glycerol molecule and with the fatty acid attached, right? With the fatty acid attached. So basically what we have to do, what these enzymes have to do, they have to break it. But why am I not drawing the line still below? Why I'm not drawing like this? Because they're not very effective in breaking down. And why are they not effective in breaking down? Because whenever you see fat, if you're washing something, some dish or something, you might have seen that that without adding detergent, you cannot just wash away the fatty, the slimy thing that is on the plate or your dish. Why? Because fat is not soluble in water, and then the bile will come into action. So we'll study then bile. Let us see what is happening right now. Right, so what is happening right now? Right now the ester bonds are being particularly broken down. Okay, the ester bonds are being particularly broken down. And so what is happening when they are broken down? Let us see what is happening. What are we getting? We are getting, there was water also, right? Remember? So that is gone, but since it is not very effective, still this fatty acid is still there, right? So, and what we get that thing that it's, right, this has broken down and right. So these are what that I have drawn. This is a fatty acid bound to glycerol. Okay, and what is this? This is free fatty acid. I'll be using FFA further. So please remember what is FFA, free fatty acid. Okay, and what is this? A fatty acid bound to glycerol. We say this is monoacyl glycerol. Okay, so this is what? This is monoacyl glycerol. Please see this reaction and please remember this. Right. So now we saw that there are still fatty acids, a lot of fatty acids, a lot which are bounded to glycerol, which needs to be broken down to free fatty acids, right? Which need to be broken down to free fatty acids. So they need to be broken down into free fatty acids. So what will happen? As we know, oil doesn't mix in water, right? So what will happen now? What will happen? Bile will come into action. The bile will come into action. We'll study about bile, what is all made up of, what is it doing. So now the interesting parts begin. Why bile will come into action? Why? Because oil doesn't mix in water. So if it doesn't mix in water, what happens? A lot of triglycerides are going in our gastrointestinal tract, which need to be broken down into free fatty acids, right? So bile is coming into action now. Let us see what is happening now, right? So let us see. This is the gallbladder. Okay. right and left hepatic duct, cystic duct, right? And here we will draw the pancreas. So we all know about pancreas and gallbladder. This is the main pancreatic duct. This is what? This is the main pancreatic duct. We have done all these things in detail, left and right hepatic duct, cystic duct. Okay, and this is what? This is gallbladder. And what is gallbladder doing? It is storing concentrated bile. Understood till now? Now let us see what is happening, right? So this cystic duct, when it meets this pancreatic duct, what happens? We remember what happens? There is a ring of smooth muscle. Okay, I'll draw it with a different color. This ring of smooth muscle, right? Remember we had studied this? Quickly tell me, what is this ring of smooth muscle? It should be in your mind, and then you should be like, oh, ma'am is telling it again, right? What is this ring of smooth muscle? This is a sphincter of Oddi or hepatopancreatic sphincter. Remember, we have studied this in detail. You can see pancreas, gallbladder, liver video, right? So now what will happen? Whenever this ring of smooth muscle relaxes, what happens? What happens? Bile comes out. What is happening? Bile comes out. And what is bile doing? Bile is an emulsification agent. And what is this emulsification agent? What is this emulsification agent? Basically, what this bile is doing? It is taking a big fat molecule and it is breaking it into what? It is breaking it into droplets. And this formation of big fat into droplets is one of the most interesting things in the whole chapter of digestion and absorption. And so let's study that interesting thing. Okay, so what is happening in duodenum? Now what is happening? We are now from the mouth, we are from the stomach, we are in the duodenum, the first part of small intestine, right? So what is happening in the small intestine, in the duodenum? There is bile and there is pancreatic lipase. Now we will study the action of bile and pancreatic lipase. And bile has many things, but for our study, the basic things that we need are: bile has phospholipids, specifically lecithin, and it has bile salts. You don't need to remember the name, but just for your interest, bile salts are there many, but cholic acid and chenodeoxy. Some people say cholic acid, some people say cholic acid. I even don't know what is the correct. And just some more information, these are derivatives of cholesterol. You don't need to remember all these things. You will study when you will do MBBS. I'm just telling for your interest. So basically bile is made up of phospholipids and bile salts and pancreatic lipase. Now the fun thing begins, right? Suppose there is a big fat molecule. Suppose, which color should I take? Okay, I'll take red. Suppose there is a big fat molecule, right? Wow, what a perfect circle. And this is like triglycerides and cholesterol, phospholipids, you know, all this we have talked about fat. And there is this, it is swimming in what? What is this? This is intestinal chyme. This is intestinal, what is this? This is intestinal chyme. So this big fat molecule, this is what? This is a fat molecule. This is swimming in intestinal chyme, but it is not able to be digested or absorbed because it is not soluble. So now what will happen? What will happen? So what will happen? Let's see. So we said that in duodenum, there is bile and there is pancreatic lipase, right? And bile is made up of phospholipid. So first, let us see what is this phospholipid going to do to our this fat molecule. So let us see, like suppose this is a, now remember that we'll be taking blue for phospholipid, right? So suppose this is our phospholipid. One side it is the ionic side, right? Ionic side or hydrophilic side, right? Now you must be understanding why this is, I am telling you specifically about phospholipid, and this is the hydrophobic side. So we can say that it is what? Amphipathic molecule, right? And similarly, we'll be having bile salts. So bile salts in purple, blue and purple. Similarly, okay, ionic side, hydrophilic side, hydrophobic side. Right, now let us see what is going to happen. So this is our fat molecule and these come and attach over here. What are these? These are phospholipids. These come and attach over here. I'll make like this. They have come and attached over here. And when they get and attached over here, what is happening? This is what side? Hydrophilic side, right? And this is what? So when they attach, now intestinal chyme, when I'm saying, I basically means the watery secretions. So now what will happen to this big fat molecule? It will be broken down into smaller molecules. And then what will happen? These bile salts, first there are phospholipids attached. I'll make a circle now. There are phospholipids attached. Okay. Now the bile salts will also come and attach themselves. What are these? These are bile salts. They will also attach themselves. So basically what? These are what? These are bile salts. And these are what? These are phospholipids, right? These are also what? Phospholipids. Okay. So you're understanding what is happening. And this is our smaller fat molecule. Smaller fat molecules, right? Now what will happen? There is this Pac-Man dude sitting over here. What is this Pac-Man dude? This is pancreatic lipase. This is still cannot work on these smaller fat molecules. And to help to aid pancreatic lipase, a protein molecule will come and get attached to its to here. And what is this now? Black thing. This is a protein molecule. Now the the pancreatic lipase is very, very happy because now it can act. It is very, very happy. Okay. Now, what will happen? Right now, what will happen? Let us see. Now you understood this. This is a big fat molecule. Phospholipids are attached. We saw the structure of like phospholipids, ionic side and ionic side or hydrophilic side and hydrophobic side. And when they are broken down into smaller fat molecules, bile salts also get attached. And then this protein molecule gets attached. Now let us see what happens. So now we have this our fat molecule, small fat molecule, right? With with phospholipids and bile salts, right? And bile salts, right? With a protein molecule. With the, now this pancreatic lipase is very happy because this is pancreatic lipase. And why is it happy? Why is it happy? Because now this protein molecule that is getting itself attached, I can tell you a little about it also. Okay, I'll tell here. This protein is also called as colipase. You understood which protein? This protein that I'm talking about. And this colipase helps pancreatic lipase to bind, and now it will have a perfect ability to break triglycerides. Okay, so just for your information, why is this protein colipase important? To get itself attached. Now these are very now this will, what will happen to these fat molecules? They will become into smaller, right? Now breaking down is happening, and these are what? These are micelles. These are what? And they are a lot of it, because now pancreatic lipase is acting on it, and there are a lot of micelles after this breaking down, right? So still we have those, right? Still we have that bile salts and phospholipids, right? Now what will happen? Very interesting thing will happen. So what are these micelles after breaking down? Let us see. Those were the triglycerides that was being broken down. So now they are into, now you must remember what is this, right? Monoacyl glycerol and free fatty acid, right? Now what will happen here? Let us see. Now there will be some few other guys who will be saying, can I hop on? Can we take a ride? So there will be some fat soluble vitamins that will say, can we take a ride on you? So now they will come, and vitamin A, vitamin D, vitamin E, vitamin K. So they will also take a ride on these micelles. Okay. Now let us see what will happen. Now let us see what will happen. Understood this much? Now these micelles are what, how should I say, they are very cool. They are very cool because they go further. They are very cool means they go further. I'll not write this, or else few students will think they are cool in temperature. Micelles go further down in the chyme, and then what? And go into enterocyte. What are enterocyte? Enterocytes, you know, we have done it in digestion. These are absorptive cells of what? Of intestine, right? So now what will happen? Suppose this is the enterocyte. Suppose this is the enterocyte. This is our molecule that we were talking about, right? This small, these micelles that we were talking about. So remember, they have few vitamins also attached to themselves. They had come and attached here. So now what will happen when it enters? It will be broken down into free fatty acids, and these vitamins will also be dissociating themselves, and some cholesterol, and right. And now one very interesting thing that I have drawn. So what will happen? Why I have deleted that? Because that bile has gone. The bile will say bye bye. So the bile salts are out now. That's why I deleted it. Are out now. And you know what? 94% get recycled and go back to liver. See, such an amazing human body we have, right? So we should take care of our human body. Anyways, all these things now, all these things will now go where? This, they will go into smooth endoplasmic reticulum. Guys, you might be thinking that I'm teaching a little different from the videos that you might be mostly seeing. Why I'm doing so? I'm doing it intentionally because I want you to enjoy the subject and do the questions correctly. Just by mugging it up, makes no fun. You can, and you will forget. If you just mug it up, you'll forget. When you'll see this video, you'll feel it like a story. I'm not saying that this is like the best video that you can watch, but yes, I have taken a lot of pains to make this video for you so that, and I enjoy that. When I'm doing that, I enjoy this because I enjoy it. When I'm doing it, I'm enjoying it. So obviously when you enjoy something that you do, the other person automatically likes to, likes your work. So I just want you to that, follow it, whatever I'm saying, and you will be able to do all the questions. I have made this, I have tried to make this, I must say like that I have tried to make this into very easy things. So let's see now again back to, they will go to smooth endoplasmic reticulum. A lot of traveling is happening in our body. So now what will happen in smooth endoplasmic reticulum? So suppose this is the smooth endoplasmic reticulum. A very interesting thing will happen now. They will be again packaged. They will be again packaged into triglycerides, vitamins, cholesterol, some cholesterol ester. Okay, so suppose this is, okay, they are again packaged into what? They are again packaged into triglycerides, vitamins, cholesterol, cholesterol ester. Okay. Now what will happen? From smooth endoplasmic reticulum, they will go to rough endoplasmic reticulum. Okay, they will go to rough endoplasmic reticulum. And what will happen over here? What will happen over here? There will be a bulge, and I'll show it with a different color. There will be a bulge like this, and they will come out. Okay, and they will come out. Okay, so they were triglyceride, lipids, cholesterol ester, and this comes out by exocytosis. Okay, and they come out into by exocytosis. And what happens? This is the first thing that happened. And rough endoplasmic reticulum puts on an outer protein on it now. So let us now add this protein. Okay, this protein. And what is this now? What is this thing? What is this thing called? Right, you will see it in many videos and books, but what is this thing called? Now this is a chylomicron. This is what is a, you can even say chylomicron or a chylomicron. And now what will happen to this? It will go to the, it will go to the lymphatic vessels or the lacteals as we call it. Okay. Now this is going to the lymphatic vessels or the lacteals. This will go. Okay. So almost we are done that, and this will go to the lacteals. And okay, suppose this is the lacteal that we have drawn. From there it will go to where? It will go to few skeletal muscles, and or it will go to adipose tissue. So what are this? It will go to adipose tissue, or it will go to skeletal muscle. And what will happen there? What will happen there? Let us see. So now one thing more. This protein was known as apoprotein because this protein is known as apoprotein. If you want to know these proteins, these are known as apoproteins. In many books it's given. And okay, so we were here. This was the lacteal and went to the adipose tissue or the skeletal muscle. Now I'll tell you very little and then it is done. So now triglycerides stored in adipose tissue constitute 98% of all body energy reserves. Okay. Why? Now the question comes, why? Why are they stored like 98% is a big amount? Because, first point, because they are hydrophobic. They are hydrophobic and they do not exert osmotic pressure on cell membranes. Okay. Right. And the second thing is that adipose tissue helps in what? Helps in insulation and protects various body parts. Okay, and protects various body parts, right? This was a little big video, but it was very interesting. And now why? Now you might be thinking that ma'am just ended it abruptly, that this goes and it goes into adipose tissue. Are ye kya matlab hai? Hai na? So now I'll tell you more about it. Now the triglycerides, because they were again packaged and sent. So triglycerides in adipose tissue are continuously broken down and resynthesized right? So these triglycerides are broken down into glycerol and fatty acid, and this is known as lipolysis. This is known as what? Lipolysis. And this is catalyzed by what? It is catalyzed by enzymes called lipases. Okay. And now one thing more, liver cells and adipose cells can synthesize lipids from glucose
or amino acids. Fun. Glucose and amino acids. Lipids can be synthesized through lipogenesis, and which is stimulated by liver, right? So liver cells and adipose cells can synthesize what? Lipids, right? From glucose or amino acids through lipogenesis, which is stimulated by very important insulin, right? And when will this happen? When will this happen? This will happen when lipogenesis happens. One, lipogenesis will happen, one thing will happen when we consume more calories than are needed to satisfy our ATP needs, our energy or ATP needs, right? ATP needs, right? Now, when the liver is low on glycogen, what will happen? If the liver, now suppose one situation, liver is low on glycogen, what will happen? It is time to eat. It is when the liver is low on glycogen, what should we do? It is time to eat. And if you do not eat, what will happen? The body starts breaking triglycerides or fats and proteins. Okay, right? So actually what happens when I'm saying if you do not eat, I mean starving, because generally what is happening normally the body catabolizes. I told you catabolizes what? Catabolizes matlab breaks down. Normally the body breaks down triglycerides and proteins, right? Normally it breaks down, but large scale breakdown will happen when we are starving. Okay, when we are starving or suffering from some endocrine disorder, then we will have this this body will be breaking triglycerides and proteins, right? So please read it once. Okay, so now one thing more, I'll just tell you the glycerol. I told you the part. So the glycerol part of triglycerides, lactic acid and certain amino acids can be converted in the liver to glucose. Where? In the liver. And these are what? These are non-carbohydrate sources. You can see these are what? These are non-carbohydrate sources, glycerol, lactic acid. And this is what is the process known as gluconeogenesis, right? So glycerol, just for an example, glycerol may be converted into glyceryl aldehyde 3-phosphate, which may be converted to pyruvic acid and which can be used to synthesize glucose. So what happened? From a what happened? From a non carbohydrate source, we got what? We got glucose, and this process is known as gluconeogenesis. Glucose is getting from a new substrata. Genesis is what? Making. So gluconeogenesis, right? Gluconeogenesis. Glucose is getting made by a new substrate, and genesis is made, right? So this is all about what? This is all about fats, and that was a very interesting session, a little big but very interesting. So this is where we started the lipids, and we did, what will we do? Chemical digestion we did, absorption, and we also did chylomicrons. So guys, kindly take notes and study. When I'm doing this, I am assuming that you have understood what I have taught you today, and I hope that it was a very, very, very interesting session. Okay, so guys, have fun and please study hard. Okay, so bye, and please